microtubule somehow. So we expect its removal from the cell to somehow affect the behavior of the microtubules, especially on the spindle. And as you can see, that's what happens. The spindle, not only is it not a nice spherical shape anymore, it's tiny and not in the center of the cell. This suggests that the spindle has somehow lost its anchor that helps to hold it in the center of the cell. This second movie is an even more extreme example. The spindle carrying the DNA is bouncing all over the cell, kind of like a mini Superman maybe. So the cell is still able to hold it in though, so it kind of just bounces. We have no idea why this happens, so it obviously suggests that the cell, the spindle has lost any attachment it has to the edges of the cell. This is interesting because it gives us a clue on how Mark may control the microtubule roles. Unfortunately, in science, it's not enough to just look at one cell and say, oh, that happens when you do this, so this must be caused by that. What you have to do is look at hundreds of cells and take a summary of what you see and try to get your results. To get this, I looked at about 400 cells maybe and divided it into two data sets, spindle phenotype, and outcome of division. As you can see, the normal cells, which is the pinky color, have mostly normal spindles, which is what you saw earlier. Dome-shaped, spherical, nicely positioned in the center of the cell with even spacing all around. Abnormal spindles, on the other hand, are the spindles that are either small, mispositioned, or just bouncing. Most of the cells with no mark have this kind of spindle, as you can see from that. Accordingly, most of the normal cells go through normal cell division, but in cells with no mark, about half of them went through abnormal cell division, where either the DNA was not separated evenly, or the cell just did not want to divide and stayed in the cell division process for a very long time, or the cell just gave up and died on me. So, yeah. But even in the supposedly normal cell division, which you can see there, so 50%, the cell often took longer to finish the cell division compared to normal cells. This is illustrated here, where I put together a frame-by-frame comparison of cell division in normal cells and cells with no mark. As you saw earlier, the normal cells all finish cell division within half an hour. But in cells with no mark, the cell takes more than twice as long to finish, which, is quite significant. You can also see the difference in spindle sizes there. It's a huge difference. But yeah, we're not really sure why this happens. Is it because the short spindles that you can see are not strong enough to pull the DNA together, to pull the DNA apart, or is it because of a different reason altogether? In the next 2 years, my project will hopefully reveal why these red spindles are short and how they affect overall forces that separate the chromosomes. At the moment, we don't know we don't know if mark plays any role in cancer. But as you can see, removing mark affects cell division, and as you know, abnormal cell division will lead to cancer. But that's not all. I think as research students, our only aim shouldn't be to just generate piles and piles of data. What we need to do is learn from these world-class research institutes that we're studying in and learn from them how to be a better researcher. Things like project management skills, using cutting-edge technology. The microscope I took this on cost £400,000. That's a lot.